All right, welcome back. So in this part, what we're going to do is start talking about some of the physiology of the autonomic nervous system, and then we're going to get into looking at some of the receptors and neurotransmitters that are involved with both sympathetic and parasympathetic, right? So to begin, there's something known as dual innervation. Dual innervation is really what it sounds like. You have two different sources of innervation for different organs, right? So in other words, the heart can be innervated by the sympathetic nervous system and also the parasympathetic nervous system. The kidneys and the stomach and the intestines and the lungs, all these things can have sympathetic and parasympathetic innervation, right? In the next slide, we'll look at some examples of different organs and what happens when they receive sympathetic and versus what happens to them when they receive parasympathetic. So you have largely antagonistic effects, but sometimes they can be cooperative as well, right? So antagonistic effects typically result in the organ experiencing two different things, right? So in other words, if you innervate the heart sympathetically, with the sympathetic division, you get an increase in heart rate. You innervate the heart parasympathetically with the parasympathetic division, you get a decrease in heart rate, right? Likewise, when you innervate the GI tract sympathetically, you have a decrease in the rate of digestive activity. If you innervate the GI tract with parasympathetic innervation, it increases the activity, right? So they generally have opposite effects. Now, why do you have opposite effects? Okay, so in other words, when you innervate the heart, why are you having a slowed heart rate when you have parasympathetic innervation versus an increase in heart rate when you have sympathetic innervation, sympathetic activity? What is the difference, right? What accounts for the difference? So maybe pause the video, try to think about why you have this difference, and we're going to move on. We're going to talk about it, right? So it turns out that the difference is because we have different receptors, okay? And these different receptors on the heart bind different neurotransmitters, right? So it's largely due to the type of neurotransmitter. And based on the type of neurotransmitter, it can bind to a different receptor. And based on that receptor type, you'll have a change in activity, right? So in other words, when you release acetylcholine onto the heart, that has an inhibitory effect. So that's from the parasympathetic division. When you release epinephrine or norepinephrine onto the heart, it has a different effect because it's a different neurotransmitter and it binds to different receptors, right? So we can look here at graphs like this and look at different glands or different organs or tissues and see what happens when you have innervation via sympathetic versus parasympathetic division, right? So just think of things easy. So we'll think of something like the bladder, right? In a sympathetic state, right? Sympathetic state, are you going to be concerned with voiding and producing urine and excreting urine? Probably not, right? So you're going to have a relaxation of this muscle called the detrusor muscle and contraction of the internal urethral sphincter. Both of these decrease the amount of urine that is flowing out of the bladder into the urethra, right? So it's slowing down the amount of urine that we're excreting, right? But if you're in a parasympathetic state, right, rest and digest, you'll have contraction of this detrusor, which is in the bladder itself, and relaxation of this sphincter. Both of those promote urine flowing out of the bladder and into the urethra, right? You can think of other things. Anytime you look at something related to the GI tract, sympathetic is going to slow it down, parasympathetic is going to increase it, right? So parasympathetic, increasing gallbladder contractions, parasympathetic, increasing secretion of gastric glands, parasympathetic, increasing smooth muscle contractions in the GI tract, right? You can do all these different things. And then you can look at, so that was increasing with parasympathetic and slowing down with sympathetic. When you look at the heart, it's exactly the opposite. So now, parasympathetic, we're slowing down the rate and contraction, but for sympathetic, we are increasing the rate and increasing the force of the contraction, right? So all of these tissues, all these organs and glands, think about it in terms of the division, so sympathetic versus parasympathetic, and what's accounting for that difference. It's largely the neurotransmitter, and it's the receptor that the neurotransmitter binds to, okay? So speaking of those, that's what we'll talk about now, these different receptors, right? In general, we're going to have two types, okay? And they're going to vary depending on the type of neurotransmitter it binds to. So we're going to have two general classes of neurotransmitters. One is going to be acetylcholine. The others are going to be epinephrine and norepinephrine, right? They're going to have similar effects. Whether you release acetylcholine or epinephrine slash norepinephrine, you'll have a different receptor. So if it's acetylcholine, you bind to a cholinergic receptor. If you bind, if you release epinephrine or norepinephrine, you bind to what's called an adrenergic receptor. All right. For cholinergic and adrenergic, you have some kind of subtypes, right? So for cholinergic, what we'll talk about are called nicotinic and muscarinic receptors. Okay, these vary based on the way that they respond to their neurotransmitter. All right, so one responds in an ionotropic fashion, and we'll talk about what this means, and one responds in what's called a metabotropic fashion. Right, all adrenergic receptors are metabotropic. So whenever we talk about metabotropic for muscarinic, cholinergic receptors, those respond in the same way that adrenergic receptors do, but obviously to a different neurotransmitter, so you're going to get a different effect. Okay, so if we first look at our cholinergic receptors, right? Cholinergic receptors bind acetylcholine, hence their name cholinergic. Okay, and you have two classes. You have nicotinic and you have muscarinic, right? Nicotinic receptors will bind a neurotransmitter, right? And they function as what's called an ionotropic or a ligand-gated ion channel. So you should hopefully recall from previous lectures the terms ligand-gated and ionotropic. This means that a ligand or some molecule binds to the receptor. When you bind the receptor then opens and it functions as a channel, a protein channel, right? So here you see a neurotransmitter, this green one, right? It's binding to this purple receptor, right? So this is serving as a receptor. So here you see it bound. And when it binds, it opens and it allows neurotransmitters to flow through, all right? So you have, allows ions to flow through. So the green is the neurotransmitter, the red are the ions. So it's ligand gated, meaning you bind the ligand or the neurotransmitter, so in this case, acetylcholine, and when it opens, it allows things to rush in, maybe sodium, calcium, whatever it may be. Okay, this is called nicotinic because they respond or they mimic the effects of nicotine that binds to acetylcholine receptors, right? So they're called nicotinic because they mimic the effects of nicotine, right? And we'll look at it again here in just a second, right? In terms of their location and whatnot. The other type is muscarinic, right? Muscarinic functions as what's called a metabotropic receptor, or they're sometimes called G protein coupled receptors, or GPCRs, right? What these do is instead of the receptor also acting as a channel, you bind the neurotransmitter, right? So just like here, you have a neurotransmitter bind. That then stimulates a cascade of events, and it stimulates something called a G protein, right? When you stimulate the G protein, you get this whole downstream, all these downstream effects that ultimately result in the opening of an ion channel, right? So now we have activated this G protein. We activate these other messengers called a second messenger. These second messengers come over and stimulate the opening of this ion channel, and the ions flow in, right? So you notice that you end with the same thing in both, right? You end with ions flowing in, you end with ions flowing in, but the way you accomplish it varies depending on if you're talking about nicotinic versus muscarinic, right? But both of these are responding to acetylcholine, okay? So if we look at these in terms of some structure, the muscarinic receptors, so the ones that we just did, we said that those are metabotropic, right? So they're responding to acetylcholine by activating a second messenger or a G protein. You can find these on target tissues that are innervated by the postganglionic neurons of parasympathetic neurons, right? So postganglionic parasympathetic neurons release acetylcholine that binds to a muscarinic receptor, right? The term muscarine or muscarine is a mushroom poison, right? And this mushroom poison mimics the effect of this whenever it binds to acetylcholine receptors. And so when you have muscarine bind to the receptor, whenever you have acetylcholine bind to these, it's going to mimic that same effect, right? So in this case, if you bind to the heart, it's going to slow heart rate, things like that, right? So you can find these on things like smooth muscle, cardiac muscle tissue, different glands as well. All right. So again, you have here you see your pink muscarinic, here you see your pink muscarinic. The postganglionic neuron, a parasympathetic, they're going to release acetylcholine um, uh, perhaps onto a muscarinic receptor at places like heart, smooth muscle, glands, things like that. Right. So if we look at nicotinic, right, nicotinic receptors, we said, are also binding acetylcholine. However, they react in a slightly different way, right? They're the ligand gated, okay? All of the um, uh, postganglionic neurons, the postganglionic neuron itself has a nicotinic receptor, right? So here's our postganglionic neuron here. On that neuron itself, so this kind of brown purple one here, it has itself a nicotinic receptor that's going to bind acetylcholine, right? So remember, both are releasing acetylcholine, right? In this case, in parasympathetic, but they're binding to different receptors, okay? So you find these in regions like the brain. You also find them in the neuromuscular junction, right? So neuromuscular junction being um, the site between the neuron and the muscle, right? So that's not going to be under autonomic innervation, but it's still going to contain this same type of receptor, right? So whenever we did skeletal muscle contraction and we talked about the neuromuscular junction, we release acetylcholine into that synaptic cleft. The type of receptor that acetylcholine is binding to is a nicotinic cholinergic receptor. Right. So remember, cholinergic being the more broad term, binds acetylcholine, and the nicotinic being the more specific type of cholinergic receptor. Okay. So hopefully you can see that right here. Now, in our sympathetic division, we're going to have different neurotransmitters, right? So all preganglionic neurons are releasing acetylcholine, and they're all binding to nicotinic receptors, right? So even in the sympathetic division here, preganglionic neuron release acetylcholine, bind to a nicotinic receptor. Preganglionic neuron um, uh, release acetylcholine, bind to a nicotinic receptor. What differs in the sympathetic division now is the postganglionic neuron, right? So if we look at parasympathetic, we're releasing acetylcholine from the postganglionic neuron. Here in the sympathetic, we're not. We're releasing epinephrine or norepinephrine, and they're going to bind to what's called an adrenergic receptor. Okay, an adrenergic receptor. The term the ADREN, adren or adreno, is referring to the adrenal gland, and the adrenal gland can also produce epinephrine. Right, so that's where we're getting this term adrenal. Right, the adrenal gland. That's where we're getting this term. Right, so the adrenergic receptor binds the hormones that can come from the adrenal gland, but they can also be released by postganglionic sympathetic neurons. Okay. So with these adrenergic receptors, they also exist in different types, right? And they're what are called alpha and beta, right? The alpha receptors usually are stimulated by norepinephrine and epinephrine, and then the beta receptors can be both stimulated or inhibited by norepinephrine depending on different factors and different variables. All right. So with these alpha and beta, sometimes you have divisions, or not necessarily divisions, but you have kind of subtypes. So you have subtypes of alpha and subtypes of beta, right? And we'll look at some of these. These are sometimes called alpha 1, alpha 2, beta 1, beta 2, beta 3, so on and so on. Okay. Remember, all of these are metabotropic. Okay, so just like those that in the parasympathetic that bind acetylcholine for the muscarinic receptor, which is metabotropic, all of our adrenergic receptors are also metabotropic. And remember the mechanism of how metabotropic receptors work, right? They activate the G protein, the second messenger, things like that, right? So there's a good mnemonic to kind of help you remember generally some of the functions of these alpha and beta receptors. And it's you use the letters A, B, C, and D, but you kind of switch the B and the C around, right? So instead of being A, B, C, D, it's A, C, B, D. A means alpha, B means beta, C means constrict, D means dilate. So A and C, if you stimulate an alpha receptor, generally you're going to get constriction. So maybe you have smooth muscle, bronchial, something like that, right? If you have beta, you're going to dilate. So maybe you dilate your bronchioles. And we'll give a an example of a medication here in a second that acts on beta 2 receptors. Okay, so if we look at some examples of alpha receptors, here we're going to have contraction. Okay, so just remember, alpha, we're constricting, or you can think contraction, we're stimulating, right? So we are contracting smooth muscle. We're going to have vasoconstriction of blood vessels. A2, we can also contract different sphincters, so in the GI tract. And then you can also prevent the release of insulin and glucagon, or I'm sorry, stimulate the release of glucagon from the pancreas. So one, you can typically think of increasing a sympathetic response. All right. For these hormones, insulin, glucagon, we'll talk about these more in AMP2. Insulin in general causes blood glucose to drop. Glucagon in general causes blood glucose to increase. All right. If we look at things like our beta receptors now, right? Beta, we said, remember beta, we said dilate, okay, or sometimes relaxation, right? Here we can have an increase in the release of saliva and the product in saliva called salivary amylase. You can also begin to break down fat tissue or adipose tissue, so something called lipolysis, where you break down triglycerides into fatty acids. You can have relaxation of the bladder. We can have tachycardia, so an increase in heart rate, increased force of our heart contraction, and then an increased automaticity, so an increase in the general rate of the heart. All right. For beta 2, we said B will dilate, so B's dilate, B and D. So you have relaxation of the uterus, decreased smooth muscle motility, so you're slowing down peristalsis. The smooth muscle is relaxing. Same thing here in the bladder, so you're relaxing the detrusor muscle. You're also going to have dilation of the smooth muscle in the bronchioles, right? And then some more effects on insulin, right? You can also dilate the arteries or the smooth muscle in the arteries and muscle and in the liver. Again, all of this is causing things to generally relax, right? And then there's one more called beta 3, and this has to do generally with fat, right? It can help break down fat and then use that as a source of heat for thermoregulation, right? So a good example of a medication acting on some of these is called albuterol, right? If you have asthma, you probably have an albuterol inhaler, or you've had an albuterol, breathing treatment, something like that. What albuterol is doing is it is what's called a beta 2 agonist, right? It binds selectively to beta 2 receptors, and when it binds to those receptors, it stimulates those receptors and then acts on the effector organ, right? So if we're talking about smooth muscle, it can cause smooth muscle to relax, right? So whether that's in blood vessels, it's in the bronchioles, the uterus, the GI tract, it's going to cause those smooth muscles to dilate. For the lungs and the bronchioles in particular, this is a very beneficial thing, right? In asthma, you have an inflammation of the airway. So the airway diameter is much smaller, it's decreased. And so you need to increase it somehow, right? And so by giving albuterol, you can have this medication bind to those beta 2 receptors. You bind to beta 2 receptors and you dilate the smooth muscle, right? Remember B and D go together, beta dilate. You dilate that smooth muscle and you get an increase in the bronchial lumen or the size of the bronchiole, so they dilate. And this is very, very good for treating asthma. Okay. All right. You can look at other things like phenylephrine that binds alpha 1 receptors that can help decrease nasal congestion. So you constrict the blood vessels in the nasal cavity and thus decrease runny noses and nasal congestion in general, things like that, right? So again, all these different medications, their mechanism of action is to bind some type of receptor. And then again, depending on the receptor that they bind, you're going to get some effect, right? You're going to have some excitatory effect. Maybe you're going to get some type of inhibitory effect, so on and so on. Okay. All right. We're going to stop here with this part, and then we will pick up in our last part by talking about somatic and autonomic reflexes. And then we're also going to kind of do an overview of both sympathetic and parasympathetic divisions.